What is CRISPR-Cas9? It's a gene editing technology revolutionizing the world because it is faster, cheaper, and more accurate. In our body, there are cells. Within their nuclei, there are chromosomes that contain our DNA, or genetic information. DNA consists of nucleobases, represented by 4 letters, A, G, C, and T, arranged in a twisted helix. Their sequence determines our unique traits, such as our eye color. If a single letter in the normal combination is switched, the DNA doesn't function properly. This leads to disorders like sickle cell or cystic fibrosis. CRISPR, an acronym for clustered regularly interspaced short palindromic repeats, is a strand of DNA used as an archive. Cas9 is a protein that can physically cut DNA strands. CRISPR and Cas9 are found naturally as a self-defense system in bacteria. Guide RNA is a single helix of nucleobases used to recognize and identify DNA strands. In gene editing, scientists take an RNA copy of the DNA they want to replicate and bind it with a Cas9 enzyme. They insert it into the cell, where it scans for every piece of DNA. When it finds a match, the Cas9 splits the DNA, damaging it. In the nucleus of a cell, there are 2 identical strands of DNA. If one is impaired, the cell uses its sister as a blueprint, copying its genetic information and repairing itself. Once the undesirable DNA is damaged, scientists add the new customized DNA. The cell repair system uses the new DNA to restore and alter the DNA's nucleobases. Using CRISPR-Cas9, we can cure diseases or grow super plants. Problem will be stopping there and not creating designer babies and developing superhuman strength.